rings of smooth muscle inside. And this is where we see a lot of the regulation that happens across the vasculature. So because of this thick layer of smooth muscle in arterioles, we can play around with the size of the radius. We can play around with the lumen, and we can play around with what? What does the radius dictate? I think I'm hearing it in the back. Resistance. The radius dictates the resistance. So because we can heavily regulate the size of the lumen and the size of the radius of arterioles, we can heavily regulate the resistance in this branch of the vasculature. All right, so what we see here is that as blood leaves the arteries, they're leaving the heart, so they're under really high pressure here. But as they move further away from the heart and into the arterioles, we see the greatest drop-off in pressure here. And because we see the greatest drop-off in pressure, we also see the greatest resistance, the greatest total peripheral resistance is present in the arterioles. And that makes sense, right? Going back to our initial relationship of flow, pressure, and resistance, in order to maintain flow, if my pressure is dropping, what has to happen to resistance? It must increase. So I'll say that again. If I'm maintaining flow and my pressure is dropping off, right, I'm moving further away from the heart, I must increase my resistance. Okay, and that's what we see here in the arterioles. We see 60% of the total peripheral resistance. We see the largest drop off. So pressure in the arteries is about 90 millimeters of mercury, and by the time it gets down into the arterioles, it's significantly lower at about 40 millimeters of mercury. And so what we see here is a lot of resistance, and we'll talk about how that can be used to maintain flow through this part of the vasculature. Now, there are three important factors that can determine the size of the radius in arterioles. We spoke about two of them already, so I won't spend too much time here. We talked about vasoconstriction, which is contracting of that smooth muscle to make the radius smaller. And we talked about vasodilation, which is decreased contraction of that smooth muscle or relaxation of that smooth muscle. And that will make the radius larger, right, allowing for more flow. The third component has to do with a concept that we saw last semester. So we talked about smooth muscle, and specifically one type of smooth muscle that is found, that is prevalent in arterioles that has this tone, right? Does anyone remember that type of smooth muscle? So one specific type of smooth muscle had this tone, this ability to contract without any external stimulus. So that was single unit smooth muscle, okay? Single unit smooth muscle. And that is what we see in the arterioles, and that is what gives them this ability to maintain this partial constricted state. So arterioles are not necessarily relaxed when they're at rest. They have this partial or partially constricted state, even in the absence of any external stimulus. So these three factors can dictate or determine the size of the radius, the tone, vasoconstriction, and vasodilation. And basically, we can determine how much blood is going to an individual capillary bed by manipulating one of these three factors, okay? So remember that arterioles are going from arteries to our capillary beds. And so in the arterioles is a really pivotal place to regulate how much blood one capillary bed gets versus how much flow another capillary bed gets. And so we can regulate the pressure by regulating the radius in the arteriole. I hope that makes sense, right? If not, we're going to try to clear it up as we go. So the way that we regulate blood flow to different organs in our body is based upon need. So think about you running away from a threat, right? You're in fight or flight mode. Which of your vascular beds, which of your capillary beds do you think should get more flow? What type of organs? Your muscles. I'm hearing muscles, right? What else? What else should get more flow to sort of facilitate you fleeing a threat? Yep. Your lungs, that would be one, yep. And more importantly, so if you think about lungs, what else are we also thinking about? Your heart, perfect. So your heart, your skeletal muscles, and then your lungs. So based upon the need of an organ, which is going to be based upon your physiologic state, we can shunt blood to and from different organs by manipulating the radius of different vascular beds. So if I want more blood to be going to my heart to facilitate that fight or flight state, I'm going to vasodilate the arterioles that are going towards the capillary beds of my heart. If I want to decrease the amount of blood going to the heart, I will vasoconstrict the capillary beds that are going to the heart. So this is what we talk about, this is what we mean when we talk about the organ flow based upon need, okay? Now, we can regulate this again by looking at our general equation. So don't forget this original equation. We talked about bulk flow being equal to pressure gradient divided by resistance. We also talked about cardiac output being equal to mean arterial pressure divided by total peripheral resistance. And here we're seeing the very same relationship, the very same equation, just in a different way. And that is organ flow, so specifically one organ, right? If it's your heart, your skeletal muscle, the flow that's going to that one particular organ will be equal to the mean arterial pressure divided by the specific resistance in those arterioles. So that very same principle we talked about in terms of the physics and the general physiology applies on the local level as well, okay? All right, so let's look at this bit of an example here. So we have the heart, and we have 3 different organ systems, and we know this side represents our cardiac output, how much flow or how much blood is going to these organs. And what we want to understand here is that the amount of blood that's flowing to organ A, let's say it's 1/3 of the circulation, and organ B gets 1/3 and organ C gets 1/3. Now, if I want to increase the amount of blood that's flowing to organ A, I am by default going to decrease the amount of blood flowing to organ B and organ C. The heart is a closed system, or the body, sorry, the vasculature is a closed system. And so if we increase the flow to organ A, we don't just magically get blood from nowhere, right? We have to take it from somewhere else. So increasing the flow to organ A will decrease the flow to organ B and organ C. Consequently, likewise, if I want to vasodilate these arterioles, I'm going to increase flow to organ A. What will happen in organ B and C? They're going to decrease, right? They're going to decrease. This is a closed system. That's the whole point of pointing this out. So if I'm increasing blood in one organ system by vasoconstricting or by vasodilating the arterioles to that system, I am going to consequently decrease flow to other areas by shunting that blood from one system to the other, right? Really important to point out. So let's look at an example of that. So we have our 2 containers, which we've been looking at, and we have a pressure gradient, right? High pressure, low pressure. Now we have 3 tubes that are representing 3 arterioles. So in arteriole A, we have a steady amount of flow, and we have 50% of the cardiac output coming here. In arteriole B, we have about 30% of the cardiac output. So right away, we know that in arteriole A, we have a low amount of resistance because we have a greater amount of flow. In organ B, we have sort of an intermediate amount of resistance because we have sort of an intermediate flow. And then we have the greatest resistance in organ C because we have the lowest flow. Okay, make sure you make that connection between the resistance and the flow, right? So if we jump to this slide and we vasoconstrict organ B's arterioles, we're going to decrease the flow that's going to organ B. So we're going to drop the flow from 30% on our previous slide to about 20% here by vasoconstricting this arteriole. So as we can see, consequently, we increase the amount of blood that's flowing to the other 2 organs. So organ A now goes from 50 to 60% of the cardiac output, and organ C is going to go from 10 or 17 to 20% of the cardiac output, right? So the point of this is we don't just move blood around and get blood from nowhere. We take the set amount of cardiac output that we're working with, which is about 5 liters per minute, and we can shift that around from one organ to the other based upon need. Now, what we're going to move on to talk about here are the intrinsic mechanisms of regulating blood flow. We're going to speak about the intrinsic, and when I say intrinsic, I mean the local regulation. So what does a specific organ do to regulate its flow? And then later on, we're going to talk about some extrinsic mechanisms. So what else can contribute to regulation? And that brings into question hormones and the neurotransmitters and so on. So let's start by looking at the intrinsic control of blood flow. So basically, local factors contribute to how much blood is coming to a certain area or how much resistance is present in the vasculature. And this will dictate the state of the smooth muscle in that vessel wall. What I mean by local factors are the state of the local tissue, right? So if you have lots of oxygen in the local tissue, then that means the tissue is well oxygenated and we don't need as much flow coming in. Right? Alternatively, if we have lots of carbon dioxide in the local tissues, then that's an indicator that we need to increase flow. And so we're going to do things like vasodilate those arterioles to bring more blood in. So we're going to move on to speak about what some of those local factors are. So when we talk about vasodilation, the factors that really increase blood flow to a specific tissue include carbon dioxide. Why would carbon dioxide increase or vasodilate arterioles? Why would the presence of excess carbon dioxide increase blood flow or vasodilate our arteriole in that area? Yep, so that we can get that carbon dioxide out and we can get fresh oxygen in, right? Likewise, excess potassium. Can anyone think of a connection between potassium and increased metabolic activity? Yeah, exactly. So the sodium potassium pump, which means that we have this sodium potassium pump on our cells. We talked about this last semester, which is pumping sodium in and pumping potassium out. Now, if we increase the metabolic activity of our cells and of our tissues, we're going to be pumping excess potassium out of the cell. And outside of the cell, in that interstitial fluid, we're going to build up potassium. So that is an important indicator of the metabolic activity of tissues. And if potassium begins to build up, that is a signal that we need to vasodilate our arterioles to bring more blood in so that we can get fresh oxygen in and get carbon dioxide and potassium out, okay? Can anyone think about a connection between hydrogen ions and increased metabolic activity? Hydrogen ions, yep. The electron transport chain is one, yep, that's correct. Anything else that makes acid? Lactic acid. What tissue do we see lactic acid in? Muscles, right? So if we have a high metabolic activity in our muscle tissues, we're going to start using anaerobic glycolysis. We're going to start building up that excess lactic acid. That's going to create an acidic environment. And that is going to be another indicator that we need to increase oxygenation, increase flow, and we can do so by vasodilating the arterioles. On the alternative state, we vasoconstrict when tissues have excess oxygen, and that kind of makes sense, right? If there's lots of oxygen in the area, in the tissues, then we don't need excess blood flowing into the area. It just doesn't make sense. It's not efficient. If we have excess oxygen, we want to kind of tell the heart to kind of slow down with the cardiac output. So we're going to close those vessels down, vasoconstrict those arterioles to kind of decrease flow because there's enough oxygen in the